And then we should share. Oh, no, it's beautiful. Please. Yes, yes. Can you see now my slides?
Yes.
Cool. So I think we can start, right? Okay. So today we will discuss associative learning, like a little bit touching again Skinner, boxes, and Pavlov, but we will try to discuss more novel approaches and just different approaches to study classical conditioning, how it could be used also in other behavioral paradigms. And then we'll discuss a paper where actually like more ethological conditions were compared with all that behaviorist approaches, and we can see that nature plays a bit different. So we'll start, I don't know what it is. We'll start with classical conditioning. So, and it seems like a bit boring. I think almost everybody heard about Pavlov dogs, and it seemed to be super simple, like, okay, stimulus, another stimulus, response. But actually, I think if we think about this a bit like broader, we can notice that all our life kind of based on this associative learning. We remember we discussed Aristotle and his like approach to think about learning as associations. So we are always building some association and try to find a connection between events in order to kind of have some predictions about the future. So it helps us to anticipate positive or negative outcomes and try to prevent negative outcomes or like do something in order to reach positive outcomes, right? Oh, no people. So hello, we started, but just started. Yes. Okay. So what is required for classical conditioning? How we define classical conditioning? In classical conditioning, we have unconditional stimulus, which is natural, and it naturally elicits some response, like food and dogs elicit salivation. And we have unconditional response, right? It's like you present a food, you see salivation in a dog. And then there is a conditional stimulus that usually neutral. What does it mean? It doesn't elicit any response because it's like not relevant for the animal. And then when we pair unconditional and conditional stimuli, conditional stimulus start to elicit some response. And in order to actually make some neutral stimulus conditional stimulus, it should precede unconditional one. So it should be a sequence of events, because if it's vice versa, there are some paradigms that utilize that, but it's usually not working. So it should be first conditional stimulus, neutral, like bell, then unconditional, like food, and then we see responses as salivation. Yes. Let's discuss a little bit fear conditioning. It's a huge field of study, and it's like important also in human research because we have a lot of studies on PTSD, which is also modeled in rodents. And actually, I worked with that a little bit. So I will show you the common paradigm, how people usually do this. So how it happens. Usually, a rodent is placed in a special chamber, and there is like conditional stimulus and neutral stimulus. It's like tone or light, but usually people use tone. And unconditional stimulus, it's aversive stimulus. It's a mild food shock. Sometimes it's not so mild, actually. If you want to model PTSD, you should apply like really good food shock. But for just anxiety or fear conditioning in a normal, not pathological way, it should be mild food shock. So, and what you do, you habituate an animal first of all, to a behavioral setup. And it's again related to our previous topic of habituation. And then you play a tone and apply a food shock. So tone should start before the food shock in order to associate it. And then test can happen like 1 hour after different intervals, and it depends on behavioral paradigm and a question, because sometimes people focused on long-term memory, sometimes like in a shorter scales. So in some time, depending on your scientific question, you test an animal, you place it in the same chamber, and like play the same tone, and then you see what animal will do. And usually a common response in this setup, we'll discuss different response in the paper later. So usually in this box, animal freeze, and freezing is a kind of behavioral readout in this case. So people quantify freezing using cameras or some infrared beams, and the more freezing animal shows, we kind of say that, yeah, animal is scared, and usually percent of time spent on freezing, it's a behavioral readout.
Can I ask you the question already?
Yes.
So in this case, you show that the conditioned stimulus is lasting for about 20 to 30 seconds.
Mm-hmm.
And the food shock or whatever you want to pair is like really like a smaller time scale.
Mm-hmm.
So is there variability depending on how long is the conditioned stimulus in a sense? Because here I see like a 20, 30 seconds is a very long thing. So it maybe like 2-3 seconds would be enough, or even more stronger actually.
There are different approaches, and I think when I did it in Moscow, we did like tone pulse. So it's like tone, like 1 second or something.
1 second.
And then short time after food shock.
Yeah.
So this is just an example where conditional stimulus was applied for like interval. It again depends on the question, and there are many paradigms, and I didn't.
So this is just an example, because
Yes,
in the classical Pavlovian thing, I think it's way shorter. Like, I mean, it's not like the bell is ringing for 30 seconds and then they wait for salivation, but or maybe I don't remember. I don't remember exactly how long was the.
Yeah, I think I put some other examples. We can see what we did. Yes.
So this is just an example.
Mm-hmm. Yes, it's not necessary for conditional stimulus to be long and to include also food shock inside of the conditional stimulus. It could be like conditional stimulus, it's finished, and also interval between conditional and unconditional stimulus could vary, because I know that people study sometimes where the difference in time between conditional and conditional stimulus too big.
Mm-hmm.
Animals cannot.
Of course, you cannot associate, because that's
so it's
too much time.
With that, people who study it in depth should play, not me. Okay, so this is examples of the chamber that usually people use for this classical conditioning. I used one on the right, and there is almost the same on the left. So usually it's like a ugly white box with a smaller box with transparent walls, and also on the floor you can see a grid, and that grid applies a food shock. So yeah, it's first of all, it's not super comfortable for animal to chill there, and also it's kind of scary and completely unnatural. And then you have these kind of dots. They have infrared beams, and also it helps to quantify freezing. Sometimes you do it with a camera, sometimes with these beams. To be honest, I don't remember what I use, but usually the setup is pretty simple. Yep. Now we will discuss different protocols, and also what should people keep in mind when we talk about fear conditioning. So there is like roughly 2 types of conditioning that could happen. So first, it's contextual fear conditioning, and second, it's cued fear conditioning. What is contextual fear conditioning? So when you habituate an animal, and then usually in protocols, you put an animal for some time in a box, and then you turn on the sound and do all this conditioning and conditioning. So this timing is needed if you want to check whether there is a contextual fear conditioning happening. Because what animal does at the beginning, it looks around and try to sample some like spatial data, spatial information where I am. And then when you, for example, here P and P, it's like a period when it's a pause, not pause, but it's a stimulus-free period. So there is no stimulus in this time. An animal just chilling in the chamber. And then you start to play a tone and also apply a food shock. And then at the end, you can actually test an animal in two aspects. So you can use the same environment, like here it's showed as a white box. You can put animal in the same white box, like day 2, day after, without any sound. And you can see that actually animal freezes. So it means that animal remembers the environment, not a cue that was cue also, we will discuss it, but even without a cue, animal freezes. And freezing here shown by a rat, like animal is scared, and after some time, this freezing kind of declines. So animal understand that, yeah, nothing wrong will happen, blah, blah, blah. And then you test animal on a cue separately. So you put animal in another context. In this case, it's a gray chamber. In reality, what we did, we just put a different color of wall, and then, yeah, animal were perceiving like, yeah, it's something different. And then you play a tone. So, and you can see that tone elicit freezing. So when there is no tone, like before, animal cannot understand that it's a dangerous environment and not freeze. And then after playing a tone, animals start freezing. So there are two components in this conditioning. It's like contextual and cued. And here just shown only for contextual one, but I think it's like when I...
So that's not correct.
Yep.
So you're showing there is a fear conditioning due to the context. So the context elicits fear behavior. And then the second one is basically both a combination of the tone and the context.
Yeah.
And either of the two elicits the freezing response. Okay, so you can maybe add multiple components to it, and every component will basically elicit this fear. So the question is, so you have on the last figure in this row, you're showing, you presented a new context that is not associated with anything, but then you present the tone in this context. And then this tone alone elicits the freezing response, right? Do they talk in this paper what will happen if you just after multiple presentations of this tone and the freezing in this new context, will this freezing will this go away if you remove the tone?
You mean...
So will this transition, will this kind of fearing behavior, freezing behavior be kind of transitioned via the tone to the new context alone?
And actually, I think nobody checked that.
I'm sure that the people checked that. I mean, people were working on that shit for like years.
More than 100 years. But
you know what's the question, right?
Yes, I understand.
Right? So can you actually just presenting the tone and the new environment, make the new environment be fearful?
I think it really depends on strength of the food shock, because actually in PTSD, we'll see it later, there is a concept of generalization. When your fear generalizes beyond these cues that you learned that elicit the fear. So even like novel context that you didn't see before and was not associated with these kind of bad situation, animals freeze. And it happens when this food shock was so bad. So it's really necessary for animal to like be super scared. And then it switches to pathological way. When PTSD, it's like pathological people sometimes scared of enclosed environments, even though they're like safe. But I think with this case, first of all, they don't show like what happens after. With this stuff, when you not apply food shock, many times this response declines really fast.
Yeah, which is not shown here, but like probably it does.
Yes. In this exact figure. First of all, it was review. They didn't show the decline, but usually after multiple representations of a tone alone without a food shock, this freezing response declines. Yes.
Yeah, exactly. It's also weird a little bit that they compare minutes to stimulus presentations, which is like one is like time domain, another one is like the stimulus, the times of how much time the stimulus was presented. I mean,
yeah, but also you can recalculate it a bit from their protocol, but usually it's how it works, because in context you just count time, how long animals spent in environment, and here you're interested in counts of tone representations.
but
That's why.
Yeah, that's clear, but I'm interested in comparing the two. Yeah, so in this case, what is a bit misleading to me is that if you look for context, yeah, so it's basically the middle figure of the right plot, so the middle. So I see that with time, the response is declining. Yeah, so without, of course, the actual shock.
Yeah, I understand what you mean.
On the right one, on the end one, I see that is not, that
it seems to be stable.
Yeah, with like I'm presenting 1, 2, 3, 4, 5, and it's still the same, which is misleading probably. It's not, it should not be the same, right? It should be of the same nature. It should decline if you don't really present the actual reason for freezing.
Yeah, it's difficult to compare.
So in this sense, it's difficult to compare. Maybe the CS presentation was, the tones were really short, I don't know, 1, 2 seconds. That's why it's just only took a minute to present 5, or even less. I don't know. We don't see the change, but that's kind of misleading in terms of learning in memory. So like the context is very different from the actual stimulus.
Yeah, it's difficult to answer the question what is more, how to say, on which cue, spatial cue, or like these kind of condition cue, animal pays more attention.
Or specific stimulus.
Yes, from this it's difficult to draw a conclusion. But we'll move on.
Yes, of course. Sorry. I think it was important to discuss.
Yes, of course. So it's like, hopefully nobody will work with conditioning, but if you will, there are some recommendations. So with fear conditioning, there are many studies made on a male mice, and now we know that it's not a good approach because fear, learning, and anxiety differs across males and females. And actually, what I like from a paper that we will discuss today, they mixed males and females, at least at the beginning. They showed like, yeah, we will try to separate them, but then they pull them up together. But anyway, this at least effort to see whether there are some difference, it's important. Then what we discussed already, protocols. First of all, it depends on your question. And second, sometimes even the same protocol in different labs works a bit differently. And actually, me, myself, we struggled with that and we didn't manage to model PTSD normally. So we failed miserably because we couldn't actually validate a protocol. So sometimes it's really difficult to understand what exactly goes wrong because it depends on animal age, animal strain. A lot of things can play a role, and we worked with old mice and they were completely like, yeah, we don't care, no PTSD, we're just chilling in the chamber. And I was like, why? So yeah, then stimulus-free periods we discussed. So it's important to give some stimulus-free period at the beginning and the end. At the beginning, if you want to see whether there is like spatial component that we discussed before, you have to give animal some time to at least understand where it is now, where it's placed. Yeah, and at the end, it's just practical reasons because you want an animal to come down before taking it. And also if you inject something just after your experiment, it's a complete nightmare. Because I was injecting my mice after PTSD, kind of PTSD model, eliciting, how to say. It was really strong food shock. And I was taking my mouse and tried to inject. And of course, it was awful because mouse was completely shocked. Mice sometimes jumped in my face. So I was like, yeah, you should give them some time to calm down. Yes. And retention tests also can vary. Retention tests, it's what we discussed before. when you test your animals a bit after the conditioning day. So depending on your question, whether you want to study long-term memory, sometimes in PTSD, you use even like 7 days interval between experiment and test because you want to see how long it lasts. So yeah, a lot of parameters that people should take into consideration. And here actually, I want to show you a protocol of PTSD that I used. So we studied PTSD in a context of pharmacology a little bit. So we wanted to see whether this drug, MK801, could prevent animal to develop from developing PTSD. So basically, it's an NMDA receptor antagonist because we know that for long-term memory, we need an MDA, and also there are different approaches. Sometimes people, really common approach to use cycloheximide and anisomycin. It's protein synthase inhibitors. So when you have LTP, long-term potentiation, there are immediate early genes that get activated in order to lead to LTP. And if you block that immediate early genes start to work, and then kind of they lead to making proteins, right? And you block that protein synthesis. So yeah, basically to wrap up, you can block memory formation by pharmacology. And here what we did a PTSD protocol. So we used 3 tones. Yes. We used
not tones, probably food shocks.
Food shocks. No, it's like 3 times you do tone and food shock, tone and food shock, tone and food shock. Here I didn't show that, but we used really short tone and then food shock, tone, food shock.
Okay, sure.
Yes.
Yeah. And it's just you say here is just you do the food shock and the contextual fear conditioning, no?
Mm-hmm.
Okay.
Yes. And it was 1.5 milliamperes. It's a lot. I tested it with my hand. It was not pleasant. Yes. And then immediately after we injected that NMDA receptor antagonist, and after 7 days, we made PT tests for, it's like a battery of the test to say that if there is a PTSD or not. And first it's a context test. You see whether there is freezing in the same context. Then sensitization test. It's kind of a bit different context. So you can see that we changed a little bit the walls, but we presented the sound. And then at the end, we used really unpleasant we changed everything. We changed environment, we put a nest, we made light a bit brighter. And in this case, environment is completely different. There is no tones. And also we changed a little bit sound environment. We turn on ventilation because it creates some sound as well. So in this case, it just, what is similar, that animal is placed somewhere, like in a small environment. And if animal shows freezing, it could be a sign of PTSD as well. Yeah. So at the end, what I can say is that nothing worked because our animals didn't show PTSD. Of course, we had a control that were injected with saline, and yeah.
So just want to ask, so like if I see in this slide the original induction of the PTSD, so which is a fear conditioning.
Yes.
So you don't show here the tone, yeah, but in sensitization test you kind of show the tone. This is called sound 75 decibel, 80 kilohertz. Yeah, so the same tone was used when you induced the PTSD, right?
I think so. I don't remember. It was many years ago.
And yeah, it's just not saying, it's just the same here, but I want to understand. So this is, you do 2 tests. One is the contextual test, and one is the tone test.
Yes.
Basically, I explained in the slide before, there's a cued test. Yeah, so you can just present the tone alone, and should also elicit it in the new context, not related to it, but it should induce the freezing behavior or something. So basically, if we say it in a more clear way, is a context test is a Q test, and this is generalization. When it's completely new environment, but that's what you Well, I don't want to waste our time and go into the detail, but 80 kilohertz is like super, super high. I know mice have like a higher hearing range, but like 80 kilohertz, maybe like don't hear. No? It's like pretty high.
I was young. It was my first normal research.
It's even hard to produce the 80 kilohertz tone.
But we did.
You need a special
in Russia
equipment.
You can.
It's very hard. So
yes. And a second.
And sorry, I'm sorry for like asking
Yes.
questions all the time, but I want to understand. So what do you mean by nothing worked? So it means like the context, none of the tests show any freezing, or what was the nothing worked?
Yes, exactly. They were not freezing.
But
First,
this means the success, right? So an MDA antagonist worked.
come on. I mean, we sometimes injected saline.
Yeah.
So in saline, that didn't work.
So
So
saline
control,
passed, so
yes.
so the controls were fucked up. Okay.
Yes, yes. And then you can say there is no PTSD. Thus, this like experiment, you cannot rely, you cannot say that there is something because control didn't work. And we can discuss in depth why it happened. First of all, our mice were old. And we were giving just fucked up mice as first, and second, like, yeah. I can add it just for example how it could be done, but how it was done, it's not so good. I hope nobody will see my video from my previous lab in Russia. Okay, then just for you, out of curiosity, maybe you're interested how it also can be, because it's like also learning and memory.
Work. I mean, why you presenting stuff that doesn't work? Interesting.
We'll see how you end up with your project. Okay. So also another thing that you can test in the using all this conditioning approach, you can see reconciliation of the memory. So you know what is consolidation? Reconciliation, right? We discussed a little bit of consolidation, that when we inject these NMDA receptor antagonist, we prevent long-term potentiation. So we assume that we prevent transitioning of short-term memory to long-term memory. It's like also really questionable approach. You can debate it a lot, but there is an approach like this, and we use that. And also, what you can do is to test to impair and interfere with reconsolidation. So for that, what you do, you do PTSD induction, like tone, food shock, as usual. Then no injection. So you kind of allow the memory to consolidate, and then you putting animal in the same environment and do what you do. You apply the tone. I didn't show it here. It's my old research, but you apply the tone. So animal remember, it freezes. And then after all this procedure, you inject antagonist. And then we assume that when we remember information, it's like you know the experiments with false memories. When you try to recall some information, memory become flexible, and then you can kind of insert new information, roughly speaking. So in this case, we try to utilize this paradigm. So when you remind animal about the event after memory was consolidated, usually in normal studies, not in our one, you can see that memory could be impaired even when it was some time ago consolidated. When we remember something, it becomes fragile. It's becoming flexible. So yeah, and then you do all the same. You want to comment? No, it's fine. It's a discussion. So we are finished with peer conditioning in this case. So if you have some questions, I can answer, because now we will switch to other approaches where classical conditioning works. No? Good. So next.
You still have people listening.
Okay. Next approach, it's condition place preference. This approach is usually used for addiction studies. So what here people can do? There are two options to build a setup. It's like a 3-chamber option and 2-chamber option. So with a 3-chamber option, animal usually placed in the center, and then it could go left or right freely. So animal can have a possibility not to choose, basically. So that's why it's called unforced choice. And another approach, animal should go somewhere because there is no 3rd chamber in between. So it's like two...
falling from the sky.
Okay. Then how this approach should be implemented? At the beginning, you take one of the parts of this chamber, like let's call it white or black, and you place an animal in a white, and all other compartments are closed, and animal is not familiar to the environment. You place it in a white, it habituates, and then at some point you inject saline or, for example, drug. And then, for example, let's say that our white compartment will be drug compartment and black compartment will be saline compartment. So you do this with the white compartment, injecting some drug, and you do the same, but with saline with the black compartment. And then in a test, you place animal in the center, and then you can see where animal goes, on the left or on the right. And in this case, we assume that animal associate environment with outcome. So, and this is how these drug addiction studies could work. Yes. Okay, biased, unbiased. Yes, here what I wanted to say, maybe it's details and it's unnecessary, but maybe some of you guys at some point will work with drugs, so you will be prepared for that. So biased and unbiased paradigm tests whether animal have some natural preference to one of the compartment. For example, you place an animal in the center without any like association, without any drug injection. And then you can see whether animal has some preferences. Usually animal goes on the left more frequently than on the right, and you can take it into account for your further analysis. So, yeah.
What is the difference between this and the previous one?
Here, animal never saw this compartment before. You placed animal here, injected with a drug, and then you placed animal here, the same animal here, injected with a saline. So animal didn't see the setup before and didn't have a chance to build some preferences, like natural preference. For example, you like pink room, but not blue room, you know? Something like this. But in this case, you see only pink room. First time in your life, you got injected. You already associated this room with pleasure.
So if I would, in this particular forced unforced, if I would just expose an animal to this whole thing before, it would be the same.
Yes. Yes.
Just adding this additional step to the protocol, like exposing to build the neutral representation.
Yes, but also you want to answer this question to yourself. So for that, you would need to do it several times. So you would also want to see not only allow animal to have a preference, but also you want to take it into account in your future analysis. So you should do several trials for that. Not just habituate animal, but also repeatedly place an animal in the center and then see where animals go.
Exactly. That's why my question, like, so the next slide, you're showing the protocol, which makes way more sense because you can have a control that the animal is preferring go to the left or right, not because of the drug.
Exactly.
But maybe there's some other reasons that the animal prefers the white one because it's more light, or the black one because it's more dark. You know, because it's the dark one is like there's a bit of a smell that's coming from the corner.
Yes.
Or something like this. So
and usually what they do, from what I read, they see if animal prefers, for example, white one more than black, they inject the black and see whether animal will change.
For example, exactly. But ideally, you have the control before that says, okay, there's no preference before, animal spends same time, same preference to go left or right, or 5 doors, whatever that is. But there's no difference. But after the injection, there's preference starts to change. Yeah?
Yes.
So.
And also, there is approach with a preference and also avoidance. For example, if you inject drug, is a preference. Of course, you wanna have a pleasure and you go to really pleasant environment to have your pleasure. But sometimes people use conditioned place aversion, where you inject some unpleasant drug like lithium chloride when animal feels sick after, and then animal will avoid that compartment of the chamber. So different approaches, pleasure or disgust. Next. So do you have any questions about this approach? No? Okay. And also, if something is too fast or not clear, you can always ask, because maybe I'm a bit messy. But we switch to these cuties. It's a taste aversion conditioning. Taste aversion is kind of special one, because usually it's one trial learning, especially in chicks. It's also studied in rodents, but I don't know, in Russia, it's like a really big thing with these chicks, because when you go to some neuroscience lecture, usually people in MSU, Moscow State University, will for sure tell you about chicks. So what they... It's not funny. It's our heritage. So how it works? There are different approaches how you can design this study. Chicks, really young chicks, they have kind of this eating reflex. When you show them a food, or even instead of the food, they show them these beads, and they start trying to eat them, even if it's not eatable. So they assume that this is food. And in this case, for example, red beets, they are covered by quinine, so it's bitter. And when chick tries to eat it, it's really disgusting, and they don't like it. But blue ones, they are fine, and they don't feel anything, even though they are not eatable. And then you're doing test. You present, you can do 3 types of the test. You can present only like bitter beets, normal beets, or mixture. And what usually people observe, that even after one trial of doing this, chicks will refuse to try bitter beets. They will be fine and will be curious again about not bitter beets. And when you mix it together, like when you put different beets all together, they will avoid red beets in this case.
I'm very sorry, but so in this case, they're really like red and blue?
No.
They're just the same. They look the same.
Yeah.
How do they look like? These beets? But so in order to understand
whether no. Actually,
yeah,
no.
because how do you know whether
No,
this
they are not red and blue. Oh, they are red and blue. Like in this paper that I read yesterday, they said that there are 2 types of beets, red beets and blue beets. And from what I heard from lectures many years ago, they're actually different colors. So chicks can
they distinguish colors?
Yes,
like
yes, they can. I also asked myself yesterday, and they can.
Okay.
Otherwise, they would not learn. Because
yeah, so they distinguish by color, not by smell, not by some other like a shape, maybe gives you an information, or no, it's just color. Because I'm curious what kind of range of their color vision is. Red and blue. No, yeah, they can. I think it's more green, blue.
There is a proof for that they can distinguish color. Do you remember the lecture about
they definitely have color vision for sure, but it's not like red, blue shaped. Yeah, but it's just a question. Yeah, I do believe that the studies.
It was a Timberger, the guy who used a big head of the bird. Remember, and they recognize a mother by a red spot under the bead. So they actually can distinguish a color, because if it's not red, if it's not the same as in the mother case, they don't care. They don't ask for food. But when they see that red spot under the beak.
So it has to be red. Are you sure? Because it can be just like a stain, of any color. So what if it's just like green, something that's close to it? Green is very close to red. So like, if it's just green, big spot, same shape, same thing, do you think they're not going to form?
Okay, let's say like this. In the paper, it was written blue and red. Yes,
no,
exactly.
I'm sure they
like
consider
this.
that, but it's just a funny point.
And then you can have another approach. So here they can distinguish taste right away. And here, for example, all beets are normal. Like they just eat beets. But then you present only green beets, for example. Chick try to eat them, like nothing happens. And then you inject lithium chloride. And it make chicks feel sick. And they was like, it's like when we eat something and get poisoned. And then they show the same behavior. So it's like whether you experience something bad with food right away or a bit after, you can learn it right away. And for, in their case, for lifelong. Yes.
Yeah. Cool. I didn't know that they have like 3-component vision. I thought it's like 2-component.
Don't pay attention. We will continue with Eyeblink conditioning.
Can I ask you something? So in the previous one, do they like also have like different color beads, like but just normal taste? So it's just like related to the colors to see if they have to control if they have any preference color-wise, like similar to the previous one.
Actually, it could be, but in this study, they didn't do that. So I don't know. Maybe they have some preference, but in this case, I think it doesn't play a big role because when you actually have some preference, for example, just imagine you have like white donut and blue donut, and you have a preference to blue because it's more beautiful. But then whether you got poisoned by blue donut or white donut, at that point, you don't care which color you prefer, because what makes you feel sick in terms of food, in this case, actually is a more important stimulus to remember comparing with just color preference. So I think in this case, in a test phase, we don't influence it much.
Yeah, that's for sure. But like if there's also a bias from the very beginning, like even though there's no difference, like if there's if there's a bias, just like I will select, for instance, blue beets all the time. And if indeed there's a conditioning to avoid red beets, then how it will be a bit biased, right?
Yes, but at the same time, by test performance, you can see whether they tried both actually, because like, for example, for you, the most important thing that in a test, they actually can show you that they still trying blue beets and avoiding red beets. Whether they tried a bit more red or blue here, I think if they avoided completely, for example, red beets, and because of their preference, they tried only blue, then they would not show you aversion in this test. So by these test results, you can kind of assume whether your initial experiment was fine or not. With blue beets, the same kind of, you can assume that, okay, they tried only red and then didn't pay attention on the blue because it was actually enough for them to kind of stop doing this strange experiment and continue their life. But then in the test, you will see whether they kind of can differentiate between these two beats, and also if they kind of keen on trying blue. So I think it's maybe in this test it could be actually important in the third one, but in the first and the second, I think, it can influence, but like globally, it will not prevent you to answer your question. Yeah, and it was already shown that they learn from first trial regarding all this food avoidance. So yeah, I don't know if I answered your question.
Yeah. Thank you.
I mean, of course, there must be always a control that the original presentation of the beads is equally, like with chambers. Yeah, so the control that we are equally taking the blue and the red, and there's no preference beginning, but then after the exposure to something, there is a preference. Again, green or blue, I mean, sorry.
Okay.
Yeah, they actually have the tetrachromats. They are better than us. So they can distinguish even ultraviolet. So sorry for noise.
No, nice to know. Nice fact check.
Yeah. So they can distinguish these colors.
So next one, and I think, no, not the last one. Eyeblink conditioning. I think before preparing for this class, I didn't read about Eyeblink conditioning a lot, but I found that it's actually widely used in cerebellum and brainstem studies. So it's more about motor conditioning. So how it works? Like before, we have air puff, and usually air puff, it's unpleasant because nobody likes when some air flows in the eyes. We kind of blinking because of that. And sometimes it's a punishment in rodent research as well. Like if a rodent performs something bad, especially in a head fixed setup, you just do that air puff and there's like, okay, I understand, I will not do that again. And then they present tone to rabbits, and rabbits don't care. You can see widely open eyes. But then when you when you condition it, like when you combine tone and air puff together, you can see that it's like a Pavlovian conditioning, because it's all Pavlovian conditioning. Exactly the same. Now tone cause eyes closure. So before it didn't, now it does. And then there are different protocols. I will say that I didn't dive in this. I just wanted to show you that this exists, and this works nicely for cerebellum and brainstem studies. And for those who are interested in that in depth, there will be some links at the end. Yes. Any questions about Eyeblink? No. Good. And the last one for Pavlovian ones, like if we think about real life, sometimes we associate, we are building more complex associations. Not only one order conditioning when you have like Bell, conditional stimulus, then unconditional food, and then salivation. Sometimes we have two or even more conditional stimuli. For example, in this figure, it's nicely shown that, for example, can opener was associated with food. When you bring something to your cat, cats usually already understand that something pleasant is going to happen. But also, if you store your food always in a cupboard, when you open a cupboard, your cat already understand that then it will be can open, and then it will be food. Of course, we can say that maybe there is a link directly between this and this, and even without can, cupboard could elicit this response. Yeah, it's questionable. There are different protocols to test this, whether this sequence actually should be in sequence or all of them will be then directly connected to kind of food. But yeah, I just wanted to say that more complex association could be built. And it also was shown in dogs in that old research. So yeah, it could be more higher order conditioning. Yeah, good. Then we will switch to instrumental conditioning or operational conditioning. And I hope we will make it like in 20 minutes or 15 minutes, and then we'll have a small break, and then we'll discuss the paper.
Before we move to this another thing, so do you want to maybe mention at some point like this aspect of the, so we are just talking about the paradigms. We're not talking about like which areas of the brain,
yes,
nothing.
Yeah, because we can, but I think it would be too much.
It would be more.
Because we have behavioral approaches to study learning and memory. Like read the heading of seminar. So operational conditioning. It's a different from classical conditioning because here you associate your actions with outcome. And here you have some control. You actually can perform action or can withdraw from performing action. And here you kind of have this willpower, more or less. And there are different approaches to implement this operational conditioning. So it could be reinforcement, could be punishment for some action, and also it could be positive and negative in both cases. So you can, if you imagine a table, like reinforcement, punishment, negative, possible, all combinations are possible. And we discussed in the first lecture behaviorists and Skinner. And here, for example, if rodent learned to perform one exact action, it receives food. And if it performs a wrong action or some different action, it will receive a food shock. And at some point, rodent will learn what is better to do, what is better not to do. Yes. And then I want to discuss common paradigms that are used in operational conditioning. This one is really interesting. I found yesterday, I knew about that research before, but I found yesterday this beautiful figure from, again, a Russian review. We have a guy, Shvurkov, and he did a lot of research in rabbits, where rabbits should press a lever in order to get food. So here, in these beautiful drawings, you can see two levers and two feeders, and they are implanted and freely moving. And we will discuss this a bit more. So what he was interested in seeing, in identifying the brain unit activity, basically with electrodes related to each step in a learning sequence. So what we discussed what he did, how the setup worked. So at the beginning, he just placed rabbits inside of the boxes, and rabbits received food from the left feeder at some time. So now actions were necessary for rabbits to perform. So they just familiarize themselves that there are feeders and that feeders can provide food. Then, since he was interested how brain activity, unit activity changes with like learning proceed, like different steps of learning, he like planned his experiments step by step. So in the first phase, he trained rabbits to perform a searching behavior when feeders stopped working by clock. Basically, when food stopped appearing at a certain time intervals, it kind of reinforced, inspired rabbits to explore the environment. So this phase was successful when rabbits actually became curious and approached the lever. They were not asked to press the lever in order to get food, but when they approached it, they were rewarded. It was the first phase. Second phase was actually pressing. So in this case, just approaching the lever was not enough, and rabbit had to press the lever in order to receive a food. And in the third phase, it was the most complex phase where rabbits had to, how to say, they had to associate that left lever lead to, how to explain?
Left feeder?
Yes, yes. And at some point they did, yes, I think they didn't do reverse learning. They had to just make left lever associate with left feeder and right lever with right feeder. Yeah. And what he did on each step, he collected data and he recorded animals while performing, so he could assess performance, how it changed over time. And what he found at the end, that it's so many. He found that neurons were active. There were some neurons that were active only during particular actions within behavioral sequence. So he found food-specific neurons that were activated when there were like food-related behavioral, when animal approached the feeder, when animal actually collected the reward. So it was like neurons that were somehow associated with food. Also, some neurons were active when a rabbit performed a lever press, and he wanted to see how changes over behavior was acquired. So on each step, he saw how these units are, how interplay between these units changes. Yes, learning.
Yeah, obviously this is like different parts of the brain, right?
Yes, he implanted in many, I know, I didn't show it, but he actually implanted in many parts of the brain. So there were different groups of animals, and they didn't have silicone probes or something, so it was really simple wires. So they needed a lot of animals. Yes. Next. So it was a bit messy, but it was like just an example how lever pressing and simultaneous electrophysiological recordings could help to answer more general questions about learning and memory. And I think we have not so many left, and I will wrap up and we will have a small break. So another approach, it's nose poking, and I will show you how this operational kind of conditioning could be used in different contexts. Like when you have an idea of operational conditioning learning, you can actually apply it to different questions in your research. For example, here there is a study about social memory, and the question of this study was to train animals to go. It was not a question, was whether animal can distinguish between conspecifics. So in the left part of the setup, implanted animal was placed. So it's a subject mice, and another small compartment of the setup was placed another animal, and it could be animal alpha or animal beta. It's not, it's just like animal A or animal B, two different animals. And our animal have to perform a nose poke sample for 500 milliseconds. So it should keep a nose inside of the nose poke hole for 500 milliseconds. Then make a choice, go on the left or on the right, depending on identity of the mouse on another side. So in this case, the global question, the global topic of this research is the social learning. But in order to have a behavioral readout, because it's really difficult to say whether animal actually distinguishes and because we cannot say without some behavioral readout whether animal learned or not, whether it can distinguish or not. Yeah, it could be like passive response when you just have one nose poke hole, force animal to nose poke for 500 milliseconds to at least like sample some data, some olfactory data from another mouse, but you want to have some behavioral readout. And in this case, you utilize this operational conditioning learning in order to answer question from completely different topic. Yeah. So, and briefly, I just briefly in this case, because we can run out of time a bit, there are more approaches to implement operational conditioning. For example, progressive ratio tasks. It's when animals asked to perform many actions in order to get a reward. And at some point, it was shown that animals have a break point. So they cannot perform too many actions. So, of course, there are different contexts, and in different contexts and different questions, this break point can be different, but in general, animal cannot act infinitely in order to get a reward. At some point, reward should be received. Otherwise, animal just give up and nothing happened. Yeah. And also there are punishment-based tasks. We discussed a little bit punishments in context of Pavlovian conditioning. But here I found an interesting approach. So it's they study decision-making more or less, and also anxiety level using punishment-based tasks. What they did, they trained animal to nose poke, and then so it was auditory cue animal should perceive a nose poke and then run back to a feeder. And then after like mastering this behavior, what they did, they sometimes associate this auditory cue with a with a punishment. It was a food shock because it's not shown here, but actually floor is a grid. So, and they varied the chance to receive a nose poke. So there are many trials in a session, and some percentage of these trials like deliver that food shock. And depending on this percentage, I don't remember the results because I was interested more about behavioral approach. They saw that anxiety level in animals differs. So you kind of anticipate that something bad can happen, but also it's not always happening, so you cannot say for sure. And it's also another approach to answer completely different question, but by means of operational conditioning. So yeah. A little bit more. Reversal learning, I will just briefly mention because it's important for almost any research, especially these tasks in context of special learning as well. For example, with this research that I showed, when mouse should perform a nose poke and then go to left or right, depending on another mouse identity on the other side, what's important is that it could be also a special learning. So it could be going to the left or to the right. It could be associated with a nose poke and with identity. So at some point, what you want to do, you want to reverse this task. So you want to change association between alpha or beta mouse with direction where our implanted mouse should go. So it kind of behavioral planning control. And also sometimes people utilize it not for control, but for checking learning flexibility, how fast animal can grasp the new rule and utilize it. Because there are some conditions when animal becomes stubborn, and especially like in the aged animals, they kind of continue doing the same things even if conditions changed. So it's like learning flexibility assessment as well. And the last in this series, it's a 5-choice serial reaction time task. It's kind of famous and it's used for assessing attention as well, because animals should go from like a base inside of the chamber and then pay attention where light, which nose poke hole was shined by light, and then nose poke exactly in that hole, and then receive a reward. So they should come here, then wait a little bit to see which spot will be enlightened. So it also kind of test for impulsivity, because sometimes animal can just run inside and then randomly nose poke and then assume that they get a reward. But for that, with that, they will not get the reward. They should be patient. They should wait until light will appear, then nose poke correctly, then go and receive a reward. So it's more complex task. And from what I heard from people, it's almost impossible to train animal to do that because it's really tough, but it's actually like nicely can model ADHD, can be used to test ADHD conditions or some attention disorders. And the last one, just briefly, it's a cool study with operational conditioning. I think everybody knows about this, but if you don't know, you should know. It's an old study where animals were implanted in different areas of reward system. And what they did, pressing the lever activated that electrodes in a reward system, and they showed that septal area made animals consistently press the lever, so they kind of delivered pleasure to themselves until death. Yes. And with that, we will make a break. I hope you were not overwhelmed, and then we will continue with paper. We will have 45 minutes, and I think it should be enough. If you have some questions, I can answer. And also, if something was not clear, you can go through slides again, because I put a lot of text and links there. And if you have some questions, you can always type me. But I think nobody will do that, actually. So, now we have 5-minute break. Are you fine with that? Good. See you in 5 minutes. What to switch off? microphone.
Maybe you want to stop sharing because someone else is going to present.
Yeah. It's fine. Okay. Do I have internet? Hopefully, yes. Okay. Catherine, I will give you access so you could share your slides. Now you should be able to.
Okay, one second. Okay, can everyone see my slides?
Yep.
Yes.
Okay, perfect. So should I begin? I'll just start.
Oh my God.
So we had a presentation. We need to discuss a paper. So just discuss a paper.
Okay, we should start.
We should. Okay. All right. So I did my paper on the ecological analysis of Pavlovian fear conditioning in rats. This paper. But I'm not able to switch the slides.
Can you try to click on them? Maybe I can.
Okay, like that.
So up and down arrows. Yeah. I don't know.
Okay. I was using left and right, so that works. But yeah, so Ava's already given us a very extensive overview of the Pavlovian model, so probably no need to run through this for probably the third time. But I did want to point out that even Pavlov suggested this model in 1897. So this has been the basis of a lot of fear conditioning research for the past 130 years. And I did want to give a little bit of an introduction into some of the other stuff that this inspired at the turn of the century, just because it's remained so influential. So the paper did mention J.B. Watson and J.J.B. Morgan and their theorizing being very fundamental to this type of fear conditioning research. And so these psychologists, they had this really interesting theory where infants were born with only 3 innate emotions. And so those 3 innate emotions were rage, fear, and love. But then they also hypothesized that this fear could only really be evoked by 2 key stimuli. So you could only truly be scared if there was a sudden noise and that terrified you, or if there was the loss of physical support. So literally, if you are dropped as a baby, like that would scare you, but nothing else would. So this implies that fear of anything else is a learned or acquired reaction. So fear was thought of as being reactional instead of largely innate. So this inspired other experiments. This was done by one of the same psychologists, John B. Watson, but then also his graduate student, Rosalie Rayner. And they wanted to show that emotional reactions can be classically conditioned in people. So they did a very similar experiment to Pavlov's experiment, wherein they showed little Albert, he's this baby, they showed him a bunch of different objects. He wasn't really scared of anything except for loud noises, confirming Watson's theory. And then they decided to expose him to a white rat and pair that with a really loud noise that was evoked by like smashing a hammer onto a pipe. And then he became conditioned so that when he saw the white rat, he experienced fear and would cry for fear of the sound. And so these sort of studies just permeated psychology for the last 130-ish years. And I mean, they're still so influential and so substantial in the field. So I thought this was sort of interesting. I was reading through a few papers this morning that were just talking about Pavlovian learning processes translationally and clinically. And so especially this one on the right that I put here, this Pavlovian learning processes in a pediatric anxiety disorders review. So in this one, there was a sentence that said something very much to the effect of people with anxiety and trauma-related disorders are thought to have Pavlovian learning deficits. So I thought that really demonstrated just how to the core it is that Pavlovian deficits are understood within the anxiety and trauma-related framework. And so then I wanted to talk about the fear conditioning in rodent models. And as you discussed, like they're typically done in these standard operating chambers wherein shocks are delivered to the animal's paws. importantly, and we'll talk about that more later in the paper. But I wanted to highlight how artificialized these environments are. And there's a reason for that. And that's because Edward Thorndike, another like turn of the century psychologist, was positing this notion of unadulterated learning. So he said that we should be studying animals' fear in an artificial environment so that we can inhibit their instinctive activities and fears, because he felt that these instinctive activities and fears might be opposite to or different from their learned ones in complex environments. So that's sort of the rationale behind that. And then I thought this was really interesting in the introduction to the paper as well. They mentioned sort of an analogous set of studies, which was called Rat Park. And this was a set of studies wherein this researcher, Bruce K. Alexander, and his colleagues at Simon Fraser University realized that in the drug addiction field, there was a lack of studies being done with rats in a naturalistic environment. And so in the paper, it described this naturalistic environment as involving trees. But I looked up a paper, or I looked up the study, and I found that they were really just paintings of trees, which I thought was kind of funny. Because you would think they would be like paintings of stumps, because obviously rats are very short, but I guess this was sufficient to be naturalistic. And so you have wood chips and enrichment toys as well. And what they found was that the rats were less likely to become addicted to a drug in a naturalistic environment, or specifically, they were less likely to self-inject morphine. And this really captured the public's imagination in the 1970s. And our authors wanted to do something relatively similar in their study. So the hypothesis that our authors were asking was, in an ethnologically relevant environment, does fear conditioning occur, and does it guide animals' future behavior? So if you guys want, you're welcome to answer these discussion questions, just because I thought this is interesting. And you may be too primed by the paper, I don't know, but I was wondering whether you would think that rats would experience fear conditioning in the wild, and whether you think that rats are more governed by fear conditioning or by instinct. Because I certainly would have guessed initially that they would be governed by fear conditioning, especially in social environments.
I think it should be both. It's as usual, like two-sided. It's difficult to say that it's only one work or another one, because it's nature versus nurture. Like it's similar, because not all threats are life-threatening. Because in case with these kind of all, of course, you want to learn from the first exposure, because you might not have second one. So you already should be equipped by like some instincts in order to be able to escape. Otherwise, you die and you'll not have chance to repeat this again. But if it's not something life-threatening, like as you said, social interactions, they are also governed by instincts, but maybe there are some aspects that you can learn, for example, where to take some food. Like just imagine a rat trying to steal something from a garbage bin, and then somebody will like, oh, shitty rat, I will kill you. And then rat will remember.
In the wild. In the wild. But at the trash bin is wild, you know.
We'll call the garbage can the wild.
I mean, it's not
a wild city.
New York.
New York, yes.
New York wild rat. So I think it's a combo.
Definitely.
Yeah, sure.
Okay, so then I'll move on to more of the methodology and the experimental setup. So something that's important to know is that these rats have all undergone surgery, and there have been wires implanted subcutaneously so that the shocks in this experiment can be delivered to the dorsal neck and back region. And this is importantly very different than the standard delivery of pain, which is to the paws. And this is part of the effort to make it naturalistic, because it would be very uncommon for a rat in the wild to encounter a predator or a human, and for this predator or human to specifically attack and harm the paws. Whereas like maybe an owl would like have its talons scraping the dorsal neck and back region. So that's making it more realistic. And then this is their chamber. So they have this nest that can be enclosed right here. They have this gate, which can be computer controlled. And then the rats can enter the foraging area, which is where they're looking for food. And these rats are hungry, and they've had food restrictions. And then there's a pellet here of 0.5 grams of food. And then this trigger zone is really important for a lot of the experiment, and it is 25 centimeters from this pellet. And then there's just a curtain here. And so the first part of the experiment is the habituation, and this lasts for 2 days, as it says. And during that part, during the habituation, they are just in the nest, and the nest is closed, and they're receiving food pellets for 30 minutes of each of the 2 days. And then the baseline trials after that, that's when they're hunger motivated and they are just exploring the arena, and there are food pellets at different increments. So sometimes the food pellet is 25 centimeters away from where it eventually will be. Sometimes it's 125 centimeters away from where it will be. It's just to encourage the rats to explore the arena during this time. And then I thought they found some sort of interesting results, and I'll kind of switch between methodology and results for the rest of this from this part of it, which was just that originally the latency to procure the pellet was a lot longer. So it took females a lot longer to procure the pellet than it did males. But then the authors just attributed this to initial spatial neophobia in female rats. But of course, neophobia, fear of something new, can't last long. So that result obviously went away. And then because that went away, they were able to include male rats and female rats in the experimental groups together. And then next, this is the main part of the experiment, maybe I would say. And so we have these 4 groups, which I can explain. And so I'll just go group by group. I thought this figure did a really good job of explaining them. So for the tone shot group, when these rats are waiting in the nest, the tone starts playing 5 seconds before the gate opens. And then the gate opens and they go and explore the arena for a variable time, however long they want, until they approach that trigger zone, 25 centimeters from the pellet. And then when they approach the trigger zone, they are shocked, and that shock lasts for 1 second. And then the tone owl group, almost the same, except for when they reach the trigger zone, there is a looming artificial owl that is supposed to look like a great horned owl that bursts out at them. And there's a demonstration of that there. Then the tone owl shock, basically the same as that, except for 100 milliseconds after the owl is emerging at them, they also receive a shock. And that's important, that interstimulus interval is 100 milliseconds, but they do vary that later. And so we can talk about the results of varying that parameter. And then there's an owl shock group, which importantly doesn't have the tone being played. So the gate just opens and then they forage and they get to the trigger zone. The owl emerges 100 milliseconds later, there's a shock. And then the tone testing we'll talk about later. But I thought it might be interesting just to watch this from the supplementary materials, just so you can see how it works. So there's the terrifying owl. The rat freaks out and he goes back to the nest, and then he's safe.
Let's watch again.
Okay. I did it.
What's that?
He's so scared. Okay, so for the results of this part of the experiment, we can look at the latencies a little bit. So the latency to leave the nest, as you'll see, the latencies were a lot longer in groups that were not the owl shot groups. So in groups that contained the tone, this is a good sign. This means that the rats can perceive the tone. It's perceptible to them. It's a salient cue. And that means that it's conditionable, which is a good sign. And then the trigger zone latencies, so the latencies to approach the trigger zone, how long that takes, about the same. And the latency to escape after the terrifying stimuli, very much the same. And then also their escape speed, there was no statistically significant difference in the escape speed after the presentation of the stimulus between any of the groups. But even though there was no difference in the escape latency and there was also no difference in the running speed, the way in which they escaped qualitatively was different between the groups. And so I thought that this would be interesting to highlight. So we'll look over here with the fear conditioning. And so in the tone shock and the tone owl groups, when they are approaching the pellet, but everyone fails when there's a terrifying stimulus, of course, these groups tend to exit linearly. Whereas in the owl shock and the tone owl shock groups, they're exiting in a really erratic and circuitous manner. And so I wish that they had included an interpretation of this just in the results as they were describing it, but it was in the discussion. And then they were hypothesizing that maybe this was because for the tone shock group, this was just a nociceptive experience. It was just painful. And with the tone owl group, it was just a scary experience. But for these owl shock and tone owl shock groups, that it was interpreted by the rats as a life and death situation. And so they panicked and consequently took these really erratic ways out of the foraging area and back to the nest. But they only set a sentence on that. And I wish that they had gone more into the interpretation because I thought that that was really interesting. But here's another visualization of the way in which they left.
Oh, it's interesting, right? I just interrupt you a bit.
Mhm.
They further will emphasize second and third group, like owl shock and tone owl shock. So I think they should perceive owl itself, even without tone, as a life-threatening experience. But then,
Right.
yes, yeah, exactly. And in this case, they are running also with a curvy trajectory. Yeah.
Actually, for me, I'm pretty bad because I just looked at the figures and I didn't read the paper. So forgive me for that. But the question is like, can you explain what is the role of the tone here? Like, I don't really understand why do they have the tone at all?
I mean, they want to see if they can be conditional, if this tone can be conditionable. So if we can later play this tone and then it will exhibit fear behavior.
Do they
If we
do
can
it
train that.
actually later on? Because all these comparisons is between the groups.
Yeah, we'll look at that later on. There's tone testing later.
There's tone testing later.
Yes. Should I continue? Okay, so the next part is the tone testing, but before they do the tone testing, they do some pre-tone baseline trials. So at this point, they're just putting the animals into the chamber, and then they're giving them maximum 300 seconds to retrieve the food pellet from here, from the nest. And so what they see at this point is between these groups, they see that there are longer latencies to procure the pellet in these owl shock and tone owl shock groups, indicating greater fear on their part because they're being a lot more cautious and wary as they go and procure the pellet. And then I thought this figure from the supplementary information was also really indicative because it shows that the tone shock and the tone owl groups did not have significantly different latencies to procure the pellet than from their preconditioning values. So this seems to indicate that these conditioning in these groups, it was not effective. And then we move on to the tone testing. So then this happens immediately after the pre-tone baseline trials. And then for these, they are released from the nest and then they come through the foraging area. They approach the trigger zone. And then once they approach the trigger zone, the tone starts playing for 60 seconds. And then they wanted to see what happened with this. So the latency to leave the nest for the owl shock and the tone owl shock groups is again longer. And the trigger zone for the owl shock and the tone owl shock groups, it is statistically significant, but I mean less so than leaving the nest latency. But as you can see here, the tone shock and tone owl animals are pretty unaffected by the tone. So that's the red and the gray. And they do end up successfully procuring the pellet, whereas these have really high averages, the owl shock and the tone owl shock group, because some of them didn't even procure the pellet, and that was just counted as 60 seconds.
But I'm sorry again. So like, as I understand, the tone was during the training, the tone was presented when they were in the shelter. And now when they test, the tone is presented in the zone.
They're going to try to investigate that as well. They're going to vary that later on. True, and they're going to present it at a different time.
This is affecting stuff, no? Right? So it's
Yeah, I would think so.
I mean, if I'm trained to always like I basically trained to associate the tone with the beginning of the trial, sort of. The beginning of the trial. It's not I don't get an association that the tone will be associated with a shock or all or whatever. It's just like the beginning of the trial. Yeah. So
right. I mean, like potentially that could just signal to you the opening of the gate, I would think, because it comes on 5 seconds before the opening.
Yeah. That's why
I thought that was odd as well.
It's odd, huh?
But yeah, we can keep going. So one of the other things that the authors noticed was that the temporal interval between the conditional stimulus and the unconditional stimulus is very crucial and particular in fear conditioning that's Pavlovian. So the authors were wondering if in fact fear conditioning had occurred, but there was a non-optimal interstimulus interval, and that non-optimal interstimulus interval was obscuring the results. So they looked at it here. This is the red is the tone shock animals. And then the gray is the tone owl shock animals. And so they're looking for an association between the interstimulus intervals and the magnitudes of the tone-induced suppression of the pellet procurement. And here, I mean, you can see there's a red triangle up here, a red triangle down here, and the R value is 0.79. There's no correlation here, which seems to indicate like a failed feared conditioning. But then here, maybe I thought even more interestingly, there is sort of a uniform tone-induced pellet procurement observed, so it doesn't matter what your interstimulus interval is. We still see that same magnitude, which I thought was interesting. And so that indicates perhaps a conditioning, but a non-Pavlovian conditioning, because Pavlovian conditioning is supposed to depend on a particular interstimulus interval. And so then here's some more of just the summarized results from the paper. And so with the tone shock animals, they're not showing contextual fear, like the fear of the arena from the pre-tone trials. And they're also not showing the conditional tone fear, as we've seen from the tone trials. And so this combination of lack of fear is suggesting that the standard fear conditioning does not occur readily in a naturalistic environment, according to the authors. And then furthermore, they say the owl shock animals, they still display fear response to the tone, even though they haven't been conditioned with it. They still escape in response to it. So this demonstrates that non-associative processes play a role in protecting animals in the wild, they say. And then one of the things that they want to do is they want to do the tone testing in the nest. So they're asking, like, is the fear conditioning really not transpiring because the tone is played when the animals are in the foraging area, like you asked? And so they switched that up, and they repeat the experiment, all of the standard procedures, but they have the tone play for 60 seconds in the nest, whereas before it played for 5 seconds before the gate entered, and then they observe the freezing behavior then. And so then they find that even after 3 tone shock pairings, the tone is not eliciting freezing and prolonging latency to foraging. So that further suggests that there's no associative learning between the tone in natural environment. And then they wanted to ask a couple other questions. So they say, is fear conditioning not transpiring because of the naturalistic chamber? So then they repeat the whole experiment, but they do have it in a standard experimental chamber, and they admit that this like introduces a couple new variables into the mix. But one of the things that they want to investigate is whether like this hunger and this food restriction is playing a part. So they have some food restricted animals, and then they feed other animals ad lib. So whenever the animals want to eat, they can. And what they find from this is that in both of the food restricted and the ad lib food groups, fear conditioning is transpiring within a single pairing in both groups of animals when it's in a standard chamber. And so from this, they surmise that there's an absence of fear conditioning, and that's unlikely to be a result of the tone qualities, because we're seeing a pairing here. And it's also probably not due to the shock placement in the dorsal neck and back region, because that was repeated here as well. It's also probably not due to the hunger restriction, because it doesn't make a difference. There's still a fear conditioning transpiring here.
Right? Like when I was reading through that, like for me, it's like I would not expect that. Before reading the paper, I would not expect that like, environment played such a big role in this case. Maybe I'm missing something, and there are some other variables, like latent variable, that we cannot take into account for some reason because we just don't pay attention on them. But why it's working in this classical setup and doesn't work in a natural conditions, for me, it's still I cannot like answer this question for myself. Like why?
Yeah, it was confusing to me at first too. I mean, I misread this the first time that I read this section, and I thought that they were saying that they had switched it to like being shocks on their paws as opposed to their neck and back, and that was causing the fear conditioning to transpire. But no, that's not the case. It was still to the same region, so it must be the environment playing such a substantial role, most likely.
What again the protocol of this constraint environment? Like, can you come back to previous slide? It was like 3 times, right? With 1 second difference, right? No, not
Sorry.
here. In this control, like with classical chamber. Yeah.
Just in this one.
Slide before.
Oh gosh.
One slide before.
Yes, sorry.
Yes. So 3 tone shock pairings. And I think it was like with 1 second difference between these tone shock pairings, right?
I think you're right.
Yeah, because I think maybe because of the protocol was not exactly the same, because what protocol they used for natural kind of environment. I remember they did 3 trials per day. Or they did it like in a row, or they did it with a bigger, how to say, time intervals between
There was
trials.
a gap, I believe, yeah.
Yes, maybe because here there is no gap. I don't remember exactly. I should check it.
There was also a difference in the interstimulus intervals. I remember that part of it with the standard operating chamber. I don't remember the specifics, but I know that that was also varied.
Okay, because it's super weird. Like, anyway, it's not supernatural. Like, anyway,
Right.
it's
Yeah.
some box. You just should run a bit more, collect food, and come back. So it's like not even close to supernatural conditions. So I would not expect such a huge difference. I don't know. I'm suspicious.
Yeah, I mean, that was one of the things that I was just going to mention, which was that this still seems highly artificialized. I mean, even comparing it to like the rat park, we have no wood chips, we have no like drawings of trees. It's just the fact that they're foraging, which they call like purposive behavior, which is supposed to like mimic, I guess, real world conditions wherein like they are hungry and they are searching for food. And then, but I mean, they're hunger restricted in the standard chamber as well. So like that's not that's not significantly different. And then they're just not foraging.
Yes, another aspect that we discussed a bit with Andrei, like in these different conditions, they utilize different strategies, right? Because they're not freezing when they see all, or when they, even without all, when there is a tone, right? They're not freezing, they're running.
No. Yeah.
And in this case, maybe because there is no place to run, and they don't have like home base where they can
nest
run, they freeze.
Right.
Also, I'm curious, like in nature, what influences these kind of choice of strategy, freeze or to run? Maybe actually having a nest influence it somehow, so you know that you have a home base where you can hide, and that's how you kind of choose your strategy, how to escape, or whether you should freeze and pretend that you're actually dead. And there is no reason.
Yeah.
It's also
and they do kind of make that point that like having them in such an artificialized environment like really does force a dichotomy of behaviors. Like you freeze or don't freeze. You don't have the option to run back to a nest, so you don't have this full like repertoire of behaviors. And so they make that as an argument for why we should be using naturalistic paradigms more.
Okay.
But yeah, it's confusing. So yeah, I was wondering like what would happen if this was an even more naturalistic environment, because it doesn't feel that naturalistic at all. Would you see like an even more noticeable absence of the fear conditioning? I'm not sure. But yeah, it is strange.
But I think those like, what do you feel about this study in general? Like, maybe it's a question to everybody. It's kind of interesting that like all these conditioning studies, it seem to be super straightforward, but when you vary conditions a little bit, you can see that everything goes wrong. Like, and it's not so simple as you would expect it to be. So yeah, and that's why this kind of nature nurture balance is important, because you want to have some control to have a readout, but at the same time, when it's super artificial constraints in behavioral setup, the behavioral is completely like fucked up. Like in this classic chamber, it's completely different response. So then in our studies, how we can take that into account? We should kind of take that into account, because a bit different setup can lead to completely different results. So it's like, yeah. Another thing that maybe just methodological thing, they have like procure pellet quantification latency. As I understand, it's like running from a home base, take a pellet and return with a pellet to home base again, right? I would, on their place, I would quantify how long does it take to go to one direction and to another direction. So I would split it to different directions because it also could vary because, for example, if it takes a lot of time for you, maybe you kind of go to arena and you're scared to go to that trigger zone in order to avoid a food shock or oval. And then on the way back, you're fast. Or it could be vice versa, that you're fast to that direction or equally fast to that in the back direction. Yes. So
I think your point about the importance of the nest is really interesting. Like if that is really significantly changing their behavior, and just because they can retreat, I don't know. I think that's really fascinating because that is a huge difference between the standard and then the more naturalistic paradigm.
So did others get the point? And maybe you have guys some other questions. We have a bit time to discuss if you wanna put a few words in our discussion, or everybody understood everybody and completely agrees with everything that was done in these studies. I cannot actually open other people. I hope others also here. Yes. So you want to say something?
No, I think I said enough.
So let's then just make a conclusion. What like in one sentence, what would you say regarding to this? Like, why it's okay, I will say something then. As I said, probably like this nature-nurture balance is really important. And maybe when we read some old studies and then like try to take it for granted, it's not completely correct. Because if there is an idea, maybe it makes sense to check it yourself with varying different parameters, like employing some more naturalistic aspects, and maybe there will be some differences like in this paper, but also this paper is questionable in some aspects as well. But yeah, I think it's nice to have this ethological comparison, at least to see that it's not like simple behavioristic approach. If you put a bit more nature in your research, you understand that it's not so easy. Yeah, it's my main takeaway that even though like over 100 years we are studying these and varying these cues like latencies and everything, it's still there are a lot of things that are uncovered just because nature is not so simple. Yeah, it would be my conclusion. Maybe it's a bit weird, but so then thank you so much. It was really cool presentation. Like you went to depth, like deeper than I asked you to go, but it's good. It's good. I even didn't have to put my words and could chill and just enjoy it.
It was election day in the US yesterday, so I wanted a distraction. So this was great.
Cool. So guys, next time we will discuss what? We will discuss motor learning. To be honest, I hate motor learning because it's not my field at all. And usually I avoided that topic during my classes. But I think it's also important to check it out because in behavioral experiments, we have some motor learning going on. And sometimes like even like nose poking is motor learning somehow and blah, blah, blah. Maybe we could also see how it influences other aspects and other behavioral paradigms, because as we discussed with habituation sensitization, like, yeah, if we think about this from aplesia point of view, boring, like whatever. But if we start to think about habituation sensitization in context of what we are doing, like different types of research, we can see that actually should take that into consideration. It could contaminate results. So I think it's important to discuss because like in a classification of learning and memory, we have motor skills learning. Okay, let's do it. I will try to dive into that and prepare some slides with some behavioral approaches. So I hope it will be interesting for me and for you, but somebody should take some paper. I don't have paper yet because I didn't search for it yet, but I think I will find something interesting. Are there any volunteers who would like to present a paper? It's not necessary to do it as cool as it was done today, so not be scared. They expect that level from you, but just put some figures, understand what's going on, and then we discuss it all together. So Georgi and Katrina already did their job. So maybe somebody else would be volunteer for the next time. Otherwise, I will suffer alone. Okay. So you should think about this. You will think?
Why not?
Okay, cool. We have one participant who wants to take this over.
Well, if no one else wants, then no problem.
Okay, guys, but next time I expect you to take something. Yep. So
the paper should not have more than 6 figures, okay? Otherwise,
you will definitely supplementary and receive
exactly.
12. Okay, so I hope it was not boring and you enjoyed this class. And I will see you in 1 week and we'll discuss motor learning and we'll try to find something inspiring from that as well. Okay, see you guys.
Bye. Thank you.